Hi everybody, welcome to our new video, and in this video today we're going to talk about DNA double helix structure. But before I start up with this video, I want to give you a little introduction. I'm from BBLC, that is Brain Based Learning Center, and on this platform we are putting videos of biology regarding the syllabus of plus 1 and plus 2, and these videos are absolutely free of cost, so you can take the benefits from these videos. And we're going to cover the whole syllabus, so it is beneficial for you if you are taking board exams or you are appearing for your entrance exam, for example NEET, right? And please do recommend these videos to people, those who need it, so that they can also get the benefit. And if you want to enroll for the full course, you can go to our social media platform and you can enroll your. So now you can see in the previous video, if you have watched regarding the nucleotide, regarding the structure of a nucleotide, we made it in the last video. So now you have an idea of how the monomer of DNA looks like, right? So in the previous video, we talk about the cell, and in every cell we have a nucleus. So this is about the cell. This is a cell, and this is inside the cell we have a nucleus, and inside the nucleus we have a genetic material, that is DNA. On this chromosome we have DNA, and when we are talking about this DNA, in last video we talk about the nucleotide. We understand how to make the structure of a nucleotide. And now we also know that this when we are making a chain of nucleotides, it will become a polymer of a DNA, right? So this whole one chain is a chain of DNA, and that is a polymer, right? So in this video, we're going to precisely talk about that how to make this chain, because in the previous video we talk about how to make one nucleotide. Now if there are number of nucleotide, how they will make a chain, in this video we're going to talk about that. Now this is the structure. In previous video we talk about that this is a pentose sugar. What we talk about in the previous video, a little recap I'm telling you. We talk about that DNA is a polymer. And what is polymer? Polymer is something which is made of monomer, that is the one smallest unit, right? So monomer of DNA is nucleotide. So to make the whole structure of DNA polymer, we need to understand that what is monomer, right? We need to understand about the nucleotide. So in the last video we talk about this nucleotide. A nucleotide is made up of three things. We learned in the last video that nucleotide is made up of three things. What are those three things? Number one is this, that is nitrogen base, right? So this is a nitrogen base. Number two, we talk about the sugar, pentose sugar. And in case of DNA, which sugar is used? We talk about this. That is deoxyribose sugar. So this is the sugar. And the third one is the phosphate. And in previous video we talk about that when this sugar and nitrogen base are made, they made a bond that is known as a glycosidic bond. And then we talk about that this sugar, when it make bond with the phosphate, the bond which is made between it is known as a phosphoester bond. So this is the structure of a one monomer nucleotide. So in this video we're going to talk about how the whole chain is made after if we are taking three monomers, four monomers, so it can be any number, right? So how the chain is built between these monomer and how the polymer of DNA one chain is made, that we're gonna talk in this particular video. So let's move on and talk about the chain, how to make a chain of DNA. Let's begin and let's make the structure of DNA double helix. So we are going to make the structure of DNA double helix structure. All right. So as we talk about that, we know the structure of nucleotide in this. First, we know this that DNA is a polymer, right? And what is polymer? Any chain or anything which is made up of monomer, that is known as a polymer, right? So it is made up of monomer. And what is the monomer of DNA? We know now it is nucleotide. So in this video, what we are basically doing, we want the nucleotide, that is monomer nucleotide, to join, right, together and make a big chain. That chain will be known as polymer, right? So this is what we want to do in this particular video. We need to understand, right? So now we know the structure of one nucleotide, right? What is the structure of one nucleotide? In previous video we learned about this. So this is how the structure of nucleotide looks like, right? This is one nucleotide. This is second nucleotide I'm making. Third nucleotide. So now we need to understand what we can do to join these monomer together and form a DNA structure, DNA chain, right? First we need to understand how to make a DNA chain. Then next we're going to talk about the helix structure, right? So these are the monomers of nucleotide. Now our question is what we will do so that they will attach and make a full chain. Now let's understand how this will happen. Now again I'm making a structure of one nucleotide. So this is C1, C2, C3, C4, and there is one C5, right? And one more. And now we know this is nitrogen and we have one phosphate group, right? So one phosphate group is there which is attached with this. And this phosphate and the bond between phosphate and this C5 is, we all know that this is known as phosphoester bond, right? We all know this. So this is a phosphoester bond. Now second nucleotide I'm going to make, C1, C2, C3, C4, and this is C5, right? And again one phosphate group is attached with this, right? So I need to make a whole chain, so I need a big space, so I'm drawing it on the next slide, right? All right, now let's begin and we will make the nucleotide and we will learn how to join monomers together and form a bond between them and make a full chain. All right, so this is the first monomer I'm making. So C1, C2, C3, C4, and we have C5, right? This is C5. And let's make this C5. We will open this structure. And now we are writing it C5 at this is C5 only, and this is CH2OH, right? And we have one OH here. Now write down phosphoric acid. And what is phosphoric acid? It is H3PO4, right? Now just open this structure and write it here. Make 4 bonds. So we have OH here, we have one OH here, OH here, and double bond O, right? So we have these two things. One is phosphoric acid, and here we have a alcohol group. This is a phosphoric acid, but it is inorganic compound. That's why you are not able to see the group COOH, right? So this is an acid only, but inorganic compound, right? And this is alcohol, right? OH group is present here, so it is alcohol. So what is a reaction between these two, right? When acid and alcohol group will react, what will happen? A esterification, right? So esterification will happen in these two groups, and what will happen? Water will be released. So from where water, what is water? That is HOH. H2O is the water. So we have to release water from these two, that is phosphoric acid and alcohol, and make a ester bond. So let's do it. So what will happen? This OH will go, and from this acid H will go, right? And this OH will go, and from this only H will go, and the full bond will be formed, right? So now let's do the esterification, and what will? Only O will left, and this is the bond which is formed in this, right? So this bond is known as ester bond, right? When water is released, then it is a ester bond. But here what we call it, what is the name of this bond? That is known as phosphoester bond, right? Why it is phosphoester bond? Because it is making a bond between phosphoric acid, right? That's why its name is phosphoester bond, right? All right, now let's make the second monomer, C1, C2, C3, C4, right? And we have here C5, right? And again, we need to make the bond between phosphorus. Let me draw it again. Okay. Now again we need to make a bond. So here we have phosphorus. Now we understand that the bond between phosphorus and phosphoric acid and alcohol is what? That is phosphoester bond, right? Now what will happen? Now we have second nucleotide. How the thing will happen in this, right? That we need to understand. Now something will happen between this carbon, that is C3, and phosphoric acid, right? Now something will happen in between these two. Now what will happen? We're gonna see it. So now so you remember what we have here. Now C3 we have OH here, right? And on C2 we only have H, because we want deoxyribonucleic acid in case of DNA, right? So in this one O is, we have released one O. I hope you remember this because we have done all these things in the previous video. So on C3 carbon we have OH group, and now the reaction will again occur between acid, that is this phosphate, and this C3 OH, right, alcohol group. Now you can imagine it's very simple. In previous thing what we did the same thing. We did a reaction between a phosphoric acid and alcohol, right? So same thing is happening here. What is it? Phosphorus and C3, what we have? Alcohol. So same reaction will occur here also, a sterification, and with this sterification this OH group will go, and from this H will go, because in esterification what will happen, water is released. So again they will make a, these two C3 and this phosphoric acid of second nucleotide will make a bond, that is a ester bond. I hope this is clear to you. One more time I'm explaining that we have two monomers, right? This is the first monomer and this is the second monomer, right? And in the first monomer we talk about it, that there is a phosphoric acid and the reaction will occur between C5, right? In the first monomer the reaction is occurring between C5 and a phosphate group, right? And the bond which is formed, that is phosphoester bond. Now what's happening here? Between the reaction is happening between C3 carbon and the phosphate group of the second nucleotide, right? So now the reaction will occur between these two. Now let's join them. So from here we, OH is gone, and from here H is gone, right? So now the bond will, now let's make the bond, and this is the bond which will occur in this, right? So now this bond is also known as phosphoester bond. Now you can see there are two bonds, right? So one bond is this and one bond is this, right? In the, when first and second nucleotide joined, there are two bonds of phosphoester, this bond and this bond. Now do you think we can call these two bonds phosphodiester bond? Yes, we can do that, right? Because in this we have two bonds. For di means two. So the name of this bond is phosphodiester bond. Let me write it. So the bond now which we made is phosphodiester bond, and you need to remember this name, that is a phosphodiester bond, because in your NEET exams or in your entrance exam this can be one question, that what is the characteristic bond of DNA, right? So what is the answer for that? That is phosphodiester bond. So what is the characteristic bond? Remember this, that is a characteristic bond of DNA, that is phosphodiester bond. So now you can understand that how this chain is formed, right? How this chain is formed. So you can join any number of nucleotide in this, maybe 2, 10, 20. So any number of nucleotide you can join, and again this kind of bond is formed and the chain is long, right? So what will, what will be the final shape of this? So let's see that. So it will be something like this. The final, how it looks like. When you're gonna join, you can say you're gonna join 3, 4 nucleotides. So how it looks like. It will look like something like this, right? Now again second nucleotide. Now let me, okay. Now again we have a phosphate group. Now this phosphate group is joined with this, right? Now again So you can join any number of nucleotide together. I join it with like this. Right? So you can join any number and make a whole chain. So this kind of chain is formed when you're joining nucleotide of DNA, right? Now, now the second thing which you need to know, that DNA chain is not a single chain, right? We cannot say that DNA is a chain which is like this chain, right? No. If we are considering DNA as a single chain, then it is not correct. DNA is a double chain, right? DNA is a double chain. So now we need to understand how the second chain is formed. As we are saying, the structure of DNA is double helix chain. So we will talk about what is helix in the next, in the next slide. But right now we need to understand that what is, how we gonna make double chain, right? So it's not tough, very simple. Same thing will happen. What we need one more similar type of chain, and we need to join them, right? So I hope this is very easy. Make one more similar chain. Let's make it. So what we need to do, we need to make one more chain. Sorry. So let's make it. So I'm making one more chain and join it. So what will happen? These two nitrogen will make bond, right? So like this, we have nitrogen here. Join it. Right, so one more. So here we have nitrogen. So here is a bond, right? So these are the bond. This is the second chain, right? Your second chain looks like this. The two nitrogen of the chain will join together, right? And make a bond, right? So this is how the double chain is formed. Now let's open this structure a little bit. I'm drawing it. C1, C2, C3, C4, and then C5, and then we have phosphate, and then we have nitrogenous base. So what we did in the when we are making these two chains, we join these two nitrogen, right? So again, I have just opened the structure and I'm making these two chains again, right? So next, the similar nucleotide I'm making, C1, C2, C3, C4, and here we have C5, and we have phosphate and nitrogen, right? But now you need to understand one thing. if we are making this kind of a chain, right? So if we are making this kind of a chain, two similar chain, this nitrogen of one monomer is on this side and the other chain monomer, other chain nitrogen base is on the other side. So how we going to join this, right? These two nitrogen are far away, and how we going to join these two, right? So now from this point we understand that DNA chain is not just similar, just parallel to each other. No, DNA chain, the second chain of DNA, let me tell you. So this is the second chain of DNA. It will move like this, right? 360, you can say. It will take a whole whole upside down, right? And it will be anti-parallel to the first chain, right? So what is the character, one of the characteristic you need to remember, that DNA chain is anti-parallel to each other, right? So they are not parallel, they are anti-parallel. So it is not like this that DNA has no head or no you can say no end, head or tail, but no, DNA chain has proper head and proper tail. You can say it, right? For example, you can imagine this is kind of a head, you can say, and this is a tail. Yeah, this is a tail. So if this is a first chain, you can call it a head, and this is a tail. If this is the first chain, how the second chain looks like? The second chain we are saying is anti-parallel, right? So the second chain is something like this, right? So here we have head and here we have tail, right? So this is, now we understand that DNA chain is what? This is anti-parallel, right? So when it is anti-parallel, now you understand, right? When it is anti-parallel, this nitrogen base of the second chain will come in front of the nitrogen base of the first chain, right? Then we can make the bond, right? It will be easy for us to make the bond. Let's see it. All right, now let's make the two anti-parallel chains of DNA and understand the structure more clearly. So I'm making the two chains, and in this structure we will understand how the nitrogen bonds are attached in the anti-parallel chain. So this is the monomer. So nitrogen is base and we have phosphate, and similarly I'm drawing the next one. Nitrogen is base and we have phosphate. Let's draw the one more nucleotide, and now we have the third one, right? Okay. Now let's join the phosphate. Let's join the phosphate of second monomer with the C3 carbon of the first monomer, right? So this is the first chain, and now what we need to do, we need to attach the nitrogenous base of the second chain, and now we understand that the second chain is anti-parallel, right? So it is like this. So we're gonna make it. So when we are saying anti-parallel, so everything is upside down, right? So let's draw it. So this is the second chain, and here we have what? We have now phosphate, right? So phosphate will be this on this side and nitrogen will be on this side, right? So now we can make the bond between these two nitrogen, right? So this is the nitrogen. So second monomer. So here we have what? We have phosphate, and here we have nitrogenous base. Now we can make the bond. Now this is a C3, right? This one is a C3, and now we can join this, right? So similarly, the third one, and here we have phosphate group, and here we have nitrogen, right? So we can make the bond between these, right? And we're gonna join these phosphate, right? Okay, so now this is the chain which is anti-parallel, and this is a two chain of DNA, right? So now we understand that DNA chains are anti-parallel, right? So this you need to remember. This is a characteristic, one of the characteristic of a DNA structure, right? That DNA is an anti-parallel chain. Now we understand that how the bond is formed between two nitrogenous bases, right? So because the chain is anti-parallel, so now we can easily make a bond between two nitrogenous bases, because now nitrogenous bases are in front of each other and we can easily make the bond. Now the bond which is formed between these two is known as hydrogen bond, right? So you need to remember this. So let me write it. So the bond between these two nitrogenous bases are known as hydrogen bond. So this is about the two chains. I hope this much is clear to you. Now the last thing remains which we need to understand. We said that DNA is a double chain helix structure, right? So the last thing remains which we need to understand, that what is the meaning of helix structure, right? So what is a helix structure? DNA chains are not just placed like this, like anti-parallel to this, right? But DNA chain are anti-parallel, and in this these two chains are coiled, right? So let me give you an example. When we are saying coil, you might have seen some circular stairs, right? Maybe in some castle or maybe in some place there are few stairs which are coiled, right? You can call it a coil, or you can call it a circular stair, right? So that kind of structure we have of these two chains. As they are coiled, right? That's why they are known as a helix structure, right? So let's draw it and it will be more clear to you guys. So let me draw it. So now we are going to understand the last thing of this topic, that is DNA double helix structure. Now we need to understand what is helix. Alright, so I hope you can imagine now what I'm trying to say. Just imagine the stairs, right? Some circular stair which you have seen. So when you are going, so it is like this, right? So coiled, or you can say a circular staircase. Now we need to make, this is the first chain, this is the second chain, right? So in this, now I'm gonna join these nitrogenous bases which will come here, right? So let me draw it. I need to make it again. Wait, I'll draw the structure again. It's just erased. Alright. So in between these two chains, I'm making a nitrogenous base. So just imagine this is a nitrogen. 1, 2, 3, 4, 5. Now I didn't write N in all the bases, but just imagine nitrogen base is present here. For example, like this, right? So it is present in all 5 bases, right? So now again I'm drawing 1, 2, 3, 4, 5, right? Okay. So now this whole from this to this whole thing is known as one term, right? Now nobody gonna confuse this thing that this half is a one term. No, this full thing is known as one term, right? So do remember this, because there are some students which those who get confused in this, that they do call this one is one term, but you don't have to do this. This whole is known as one term. This is a question of your entrance exam. Sometimes they ask that what is one term, right? So this one term is also known as the pitch or DNA, right? Remember this, they do ask what is the pitch of DNA. So this one term is known as pitch of DNA also, right? Okay, so in this one pitch, what is the length of this one pitch? So do remember this, the length of this one pitch is known as 3.4 nm, right? Or you call it a 34 angstrom also, right? So this is the length of a pitch, right? And this length of a pitch, how many, you need to remember how many bases we have, how many base pairs we have, you need to remember this, right? So in this whole one term we have 10 bases, and 20 base pairs, right? So you need to remember this. So now we said that there is a turn of, we just said that there is a turn of 360 degree. Now they may ask, now I'm talking about this turn, right? They say that there is a 360 degree. Now what is the angle between two base pairs, right? They may ask you what is the angle between these two base pairs. So this is very simple. Now how many bases we have? We have 10 bases. And now in this whole, what is the angle? That is a 360 angle. So 360 divided by 10, what we get? That is a 36. So what is the angle between this is? That is 36 degrees. So between two base pair, what is the angle? That is 360 degree is the angle. So this is what you need to remember regarding the double helix structure. And the structure which I have drawn is a BDNA, right? There can be other DNA also. So the structure which we just not talk about is a BDNA, right? So this is all in this particular topic which we talk about. So this much is in your syllabus. So now just I want to recapitulate